Okay, so picking up where we left off in class, we're going to talk about the cell cycle and what actually controls the cell cycle. So we discussed how different cells divide at different rates. And some cells, for example, like our intestinal cells seen here, are rapidly dividing. In fact, they are dividing probably you completely replace your intestinal cells every 5 to 6 days. So that's a very rapid rate of division. In class, I showed you the video of the C. elegans embryo, which is so rapidly dividing that it skips G1 and G2. So it goes straight from S phase to mitosis to S phase to mitosis. Other cells in the body, in fact, the majority of the cells in our body are not actively dividing at all. And those cells are actually in G0. So examples of cells that are in G0 include our neurons and our skeletal muscle cells. So these are considered to be post-mitotic or non-cycling cells. So how do all of these different cell types know when they should divide and when they should stop dividing? Let's take a look at how this cell cycle control is regulated. Some of the early experiments were done in the South African clawed frog, which is shown here. It's called Xenopus laevis. And part of the reason that this organism was used is that it has very large oocytes or eggs cells that are very useful for biochemical analysis. So some of the earliest experiments included some sort of mixing and matching experiments where scientists would take the cytoplasm out of an egg that was actually in mitosis or M phase, and they would inject that cytoplasm into an egg that was in interphase. Okay, as a control, they would take interphase cytoplasm, so cytoplasm out of an egg that was in interphase, and also inject a cell that was in interphase. And the question here was, is there something that's actually in the cytoplasm of the cell that can cause or control whether or not a cell goes into mitosis? And so what they found was very interesting. When they did this, injected the cytoplasm, they found that the cells that had been injected with the mitotic cytoplasm immediately entered mitosis. So the nuclear envelope would begin to break down, the spindle would begin to separate, and the chromosomes would begin to condense. And then with the cell that had received the interphase cytoplasm, it stayed in interphase. And so this experiment suggested that there is something within that cell, and specifically there's a factor that's actually in the cytoplasm that can promote a cell's entry into M phase or into mitosis. And they called this factor, let's get the pen here, the MPF, M phase promoting factor. Okay, so at that point did not know whether or not this was a protein or some other signaling molecule, but they knew that there was something inside that cytoplasm that could induce a M phase or an interphase cell to go into M phase. So what exactly was in this MPF? What was it made out of? The MPF is a complex of two different proteins. So it's comprised of two components, and both of these are proteins. The active component here is what's called the CDK, and that, let me write that in the blank here. There is a protein kinase called CDK that actually stands for cyclin-dependent kinase. And so this, in class, we've discussed kinases when we talked about the cell signaling, and kinases are responsible for phosphorylating cells or adding a phosphate group to cells typically to activate them or to change their localization or some other function. So this was the sort of active component of the complex, and then there was a second protein that was called cyclin. Okay, and cyclin is a regulatory molecule that controls the activity of the kinase. So let's write cyclin here. And it's only when the cyclin is actually bound to the CDK that this whole complex can actually phosphorylate other proteins. So it's not active unless it has both components, both the cyclin and the CDK. Well, the cyclin, it turns out its name comes from the fact that this protein actually cycles, or it varies in concentration in the cell. So when scientists actually analyzed cells and looked at what happens to CDK throughout the cell cycle and what happens to cyclin throughout the cell cycle, they had sort of a surprise. So they were surprised to find that the MPF CDK, let me get the pen, stayed at a constant level throughout the cell cycle. So the MPF was constant at a constant concentration, but what varied or what cycled throughout the cell cycle was the levels of the MPF cyclin. And what was very interesting is that through G1S and G2, the amount of cyclin was increasing, and once it reached a threshold here, it formed the active MPF complex, and that complex could then activate a number of proteins by phosphorylating them. Okay. What was interesting then too is that they observed that after M phase, that cyclin was destroyed. So the levels of the MPF cyclin decrease at the end of each M phase and then again increase in the next round. So there's sort of this oscillation in concentration of the cyclin portion or the regulatory portion of this molecule. Okay. And this is actually not just true for the M phase CDKs, but it turns out that there's other CDK proteins in the cell as well. So to go into a different phase of the cell cycle, there are different CDK cyclin complexes. And you don't really need to know the names of these, but just the important point here is that in addition to the fact that we do have this M cyclin, oopsies, there are other cyclin CDK pairs that regulate entry into S phase. So the G1 to S CDK complex regulates that transition between G1 and S. There is an S phase cyclin CDK complex that phosphorylates the targets that you need for S phase. And then, of course, we have the MCDK. So each of these different CDK complexes will cause the cell to actually specifically enter that particular stage by phosphorylating the targets that you need for that stage. So since we've talked a little bit about the MPF, what are some of the targets that might be phosphorylated by the MCDK? Well, some of the things that happen in mitosis, we have the chromosomes condense, the nuclear envelope has to break down, right? The spindle has to form. So if you look actually at the targets of this MCDK complex, I'm not surprised to find that these are the targets of the MPF. It phosphorylates proteins called condensins. And these condensins are proteins that help to cause the chromosomes to condense down. It phosphorylates proteins like lamins, components of the nuclear pore complex, and internuclear membrane proteins. All of these proteins help the nuclear envelope to break down, which happens very early on in prophase. And then you might imagine that you need to sort of separate the different organelles. And so another one of the targets is Golgi matrix proteins that helps the Golgi to fragment so that you can partition it into the two daughter cells. And finally, of course, we need to form the spindle. And so there's centrosome proteins as well as other microtubule associated proteins that are targets of this MPF so that you can facilitate the formation of that bipolar spindle. Okay. So this system of CDKs can sort of indicate to the cell when it's ready to enter the next phase of the cell cycle. But of course it has to make certain checks. It has to check certain things before actually entering into a particular stage. So there are a series of checkpoints. And I have here a picture of a washing machine because this is very analogous to a washing machine. When a washing machine runs its cycle, it has various steps, but before it goes to each step, it checks to see that the last step has been completed appropriately. So if you think about it, before the spin cycle, the machine checks to make sure that all of the water has drained, and then it begins the spin cycle. etc. And you can even, in your washing machine, you can hear it click when it's going from cycle to cycle. And so with cell cycles, the cell cycle is very similar, right? There are checkpoints that have to be passed before a cell can go from M phase to G1 to S to G2. So we're going to talk about three main checkpoints here. The first of which is called the M phase checkpoint. This is going to be a checkpoint to make sure that each set of chromosomes has attached to the bipolar spindle. So in other words, the sister chromatids have bipolar attachments. Each kinetochore is attached so that when you go to anaphase, you can pull those two apart. There's another checkpoint. We'll talk about some more details of these. The G1 checkpoint is going to check for cell size, check for damage, check for nutrients. G2 is going to check to make sure that once S phase is over, that you've actually replicated all of the chromosomes accurately and check for damage before you go and divide the cells in M phase. So let's look at some of the details of these. The M phase checkpoint, as we mentioned, is going to confirm that all the kinetochores have attached. So just to kind of draw this out, when you've got a cell that is in, hard to draw with this little pen, is in, let's say, metaphase. So we've got the bipolar spindle, and we have the chromosomes lined up along the metaphase plate. And let's just imagine, I think I can only fit two chromosomes. So when the cells are making their attachments, they should actually have, each chromosome should have a bipolar attachment, meaning that it's attached to centrosomes at each of the poles. So imagine for a second that the cell is in the process of forming this attachment, but doesn't quite get this chromosome attached to this centrosome. If the cell allowed the chromosomes to begin separating before all of these bipolar attachments have been made, what's going to be the consequence? Well, you can see what's going to happen here is that, let's draw out what's going to happen. We're going to create one cell that has one sister chromatid for this chromosome. And down here, it's going to actually have two. And you're going to create a second daughter cell that just has one sister chromatid. And it's missing the second chromosome. And so this, of course, is a problem for the cell. This is what we call aneuploidy. So let me write aneuploid. Aneuploidy means the wrong number of chromosomes in the chromosome set. This can lead to things like Down syndrome if it happens during the formation of the sex cells. It can also lead to cancer if this occurs during somatic cells or within the somatic cells. So this step here, checking to make sure that each one of these chromosomes is attached, is critical to make sure that we don't get things like aneuploidy. Okay? We'll come back in a second and talk about the details of that. But first, the other two, as we mentioned, the G1 to S is going to check to make sure the cell is large enough. It has to check and make sure it's got enough nutrients and the signals that it needs to divide. So it's only going to enter into S phase and commit to dividing if it's got the necessary nutrients. The second, of course, set here is going from G2 into M phase because that's, of course, the committed step to divide into those two cells. And so first it has to check to make sure all the DNA damage has been repaired, if there's any DNA damage. And then also it has to check to make sure that that cell is large enough. Remember, one of the major things that happens in G2 is that the cell increases in volume because it's got to create two identical daughter cells at the end of this process. Okay, we're going to talk a little bit about this first checkpoint. And so you will need to know the details of the M phase checkpoint. We're going to talk about what proteins are involved in this checkpoint. This checkpoint involves a number of players. So let's look at the players that are involved. There are proteins called cohesins. And these proteins, much like their name suggests, if you think of cohesive sticking together, the cohesin proteins keep the two sister chromatids together. And if you look in the picture here, here's cohesin. Let me get the pen out. This is the cohesin, and the cohesin, if you look at where they are, they're holding the two sister chromatids together. Okay, so they are going to be there until the cell is ready to go into anaphase. They are going to be holding those sister chromatids together. So who are the other players in this pathway? Well, you've got an enzyme called separase. We know it's an enzyme because it has ACE, separase, and as the name implies, this is going to be the enzyme that actually cleaves the cohesin protein. So in this schematic, it's shown as scissors. So this protein, when it becomes active, it won't be active right away, but when it becomes active, it's going to go through and snip, 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 cut these cohesins apart so that you can enter into anaphase. However, as we said, separase is not active right away. It's got to wait to make sure that all of the chromosomes are attached to both poles. And so there's a protein that prevents the activity of separase, and that is a protein called securin. So you can see securin here basically forms a block that holds those separase enzymes inactive. And it does so until you've checked to make sure that every single chromosome is attached. Okay, so how do we then release securin? Once we do have all of our chromosomes attached, how do we release securin so that separase can do its job? Well, this actually, we have a nice little space here to draw our cell. What happens is that, in this case, we'll draw 4 chromosomes. As these chromosomes are getting attached to the spindle fibers, here's one centrosome, here's another centrosome. Let's put the microtubules are beginning to attach here. So as they're attaching, if there is a chromosome that is not attached yet to a spindle, so we'll do again this last one, there's a number of proteins that are part of the spindle assembly checkpoint pathway. And so we call these SAC, spindle assembly checkpoint proteins. And they send out, if this chromosome is not attached, they send out a very loud signal that says, I am not attached. It's a negative signal. As long as these spindle assembly checkpoints are there and signaling, the cell, the securin will prevent the activity of separase. So that securin will be there. So now once this one, oopsies, oh, now my thing is gone. Now I have to draw this back in really quickly. Once I do get all of the, quickly draw this thing back in. Once all of these 4 chromosomes are attached, there's no more negative signal. Okay, and I'm not even showing my chromosomes attached very well, but so once all of these are attached to the spindle fiber, spindle apparatus, there's no more negative signal. And once there's no more negative signal, once there's no more negative signal, this inhibitory arrow is gone. So basically these spindle assembly checkpoint proteins are inhibiting a protein called APC. Now APC stands for anaphase promoting complex. So that tells you exactly what that complex does. When it's active, it's going to promote anaphase, or the separation of the chromosomes. So anaphase promoting, I need a different tool for writing. Complex. So once those spindle assembly checkpoints are gone and no longer sending the negative signal, the anaphase promoting complex becomes active. Okay, there's no more inhibition of this. So it becomes active. What it does is it attaches ubiquitin, which are these little UB, that stands for ubiquitin. And ubiquitin is a small protein that signals for the degradation of whatever protein it is attached to. So when you, sorry, this is an I right here. When it gets attached to securin, it leads to the degradation of securin. So now that securin is gone, chewed up, then the separase can become active, and it can actively cleave the cohesin. So once it becomes active, it cuts all of these cohesin pieces apart, and these chromosomes can separate and go to the opposite poles. Okay, so I hope that makes sense. If you need to go back and kind of watch the steps again, the main point is that these spindle assembly checkpoint proteins are sending negative signals until all of these microtubules are attached. And then when these are gone, the APC or anaphase promoting complex can become active. And its job is going to target securin for degradation. And how does it do that? It tags it with these ubiquitin proteins, and that sends the securin to the proteasome, which chews up the protein. That releases the separase, and then the separase can cut all of the cohesin proteins so that the two chromosomes can separate to opposite sides. Okay. So how do they get pulled to opposite sides? When we show in an anaphase, we just show the chromosomes kind of pulling apart. What's actually happening here is you essentially have all of the spindle fibers, the microtubules that are attached to the kinetochore. So remember we said that there's the DNA region right here. This DNA region, that's called the centromere. Okay, so this is the centromere. And the DNA is attached to proteins that are part of the kinetochore. So those are the complex of proteins, about 100 different proteins that are sort of the mediators between the DNA, the chromosome, and the microtubules. So this, in this electron micrograph, you can see these microtubules attaching to that kinetochore plate. And when they pull them apart, the kinetochore attaches to the microtubule, and then that microtubule shortens right at this end. So as it gets shorter, because the kinetochore is hanging on to it, it actually pulls it apart. And so then if we look in a cell, what this looks like is basically those microtubules are pulling, oopsies, I didn't mean to do that, but so the kinetochore is pulling on the centromere, and the microtubules are getting shorter right here, and as they get shorter, they pull the chromosome with it to each of the poles. So they're shortening from the plus end of the microtubules and pulling them apart. Okay, and that allows you to segregate your chromosomes. All right, so that's the M phase checkpoint, and you should know those players. You should know the spindle assembly checkpoint proteins, APC, securin. You should know separase and the cohesins. Okay, so coming back to the checkpoints again, let's talk about how you get into S phase. If you're in G1 and you're ready to enter into S phase, have to check a couple of things, and then you're going to go into S phase and start phosphorylating all of the components that you need in order to replicate the DNA. So let's look at this G1 to S checkpoint. This, of course, is going to involve a different CDK. So this is the CDK for specifically for S phase. And in this sort of regulation pathway, the final target protein that is going to get activated is a protein called E2F. Okay, and so E2F is a transcription factor. So it's going to turn on genes. It is going to increase the expression of genes that are needed for S phase. And so what are some of these genes? They're going to be genes that are involved in DNA replication and in centrosome duplication. Okay, so these are your S phase genes. So they can be for DNA replication. I'll just write DNA rep. And they can be for centrosome duplication. Okay. But before you become active, this E2F protein is maintained in an inactive state. And the protein that binds to E2F to keep it inactive is this protein right here, retinoblastoma. So I have the name written out right here, retinoblastoma, or RB. is a protein that will bind to physically inhibit the E2F protein. So let's look at how we release this E2F transcription factor so that it can go and phosphorylate, not phosphorylate, it's going to increase the expression of the genes that we need for S phase. Okay, so how this works is that the S phase CDK that's paired with the S phase cyclin, this is activated. So that basically the concentration of the cyclin increases. There's an inhibitory phosphate group on that CDK, and that inhibitory phosphate actually keeps the CDK inactive. So that inhibitory phosphate will be removed, and that will activate this whole S phase CDK complex. Once that's active, it's going to phosphorylate a target. So what is the main target of this CDK? It phosphorylates and alters the conformation of the retinoblastoma protein. So this RB protein is one of the targets for the S phase CDK. When that gets phosphorylated, it changes conformation, and so it can pop off of the E2F. And that, of course, releases the E2F to go turn on genes that it needs for S phase, DNA replication genes, as well as centrosome duplication genes. Okay. So interestingly, along with a lot of these checkpoints, there are things that can modulate the checkpoints. And so, in fact, there are certain viruses that mess with those checkpoints. And one example that I wanted to talk a little bit about is the human papillomavirus. The human papillomavirus actually produces a protein called E7, and this E7 protein is able to bind to retinoblastoma. So even in the absence of the signal to divide and the activation of the SCDK, this E7 protein actually binds to retinoblastoma and gets it to release E2F. And so what's the effect of that? You have E2F is released, so E2F is going to induce S phase. And so this is essentially an unscheduled S phase, and it's going to get those cells to enter into the cell cycle before they're actually ready to do that. Okay. The reason the virus wants to do that is that the virus needs to be replicated itself, and so it wants the cell to go into S phase so that it has that machinery, that host machinery that it can take advantage of. Okay. So many viruses have kind of these interplays with the cell cycle, so they can mess with our cell cycle. And in fact, you may have heard that HPV can cause cancer. And so HPV does, in fact, increase the risk of certain types of cancer, and in particular of cervical cancer, by inducing those unscheduled S phase entries, so inducing those cells to divide. If you look at an infected cell, it begins to divide rapidly. And for most patients that have HPV, about 90%, will heal within 2 years. So they can actually get rid of that infection. But some will actually develop cancer. So roughly 0.8% of individuals where the HPV DNA gets integrated into the genome of these cervical cells can develop cancer. You probably heard about Gardasil, which is a vaccination that prevents some of the most harmful forms of the virus, and so that is now available. And they encourage young people to have this because many people actually do get HPV infections during their lifetime. Okay, so I'm going to stop right here, but our next topic is, of course, cancer. So I will start a new lecture for this second portion just to keep these lectures a little bit shorter.